Bismillah Rahman Rahim. So welcome back friends. Welcome to another video lecture of this series which regarded to DNA transcription in prokaryotes. Okay. In the previous video lecture of this series of DNA transcription, we talked about the protein, we talked about overview, we talked about the initiation of DNA transcription, and we also talked about the elongation of DNA transcription. Now this is the last video lecture regarding to DNA transcription in prokaryotes, and in this video lecture we are going to talk about another stage of DNA transcription. We call that termination of DNA transcription. Okay, now how the termination of DNA transcription are going on in case of the prokaryotes, and what are the main proteins are involved in this case, we'll be talking today in this video lecture. But before that, you need to understand the basic concept about the DNA transcription, and you need to understand initiation and elongation as well. If you missed this video lecture, it is present into my channel. You can go back and watch this video first, and then you will get idea about the termination of DNA replication, DNA transcription in a detail. Okay, so let's clean the whiteboard and let begins the topic. Okay, so let me write termination of DNA transcription in prokaryotes or mainly in E. coli cell. First of all, what is termination of DNA transcription? The termination of DNA transcription is that stage in DNA transcription when a long RNA strand are synthesized during this process, and after that, when this RNA strand are get separated from between the DNA strand, and then this RNA are further modified. So this is called termination of DNA transcription. Means at that stage the RNA are completely synthesized and it will be dragged out from between the two strands of the DNA. Now how the whole process will be going on? Remember, in case of the prokaryotes or in case of E. coli cell, we have two types of the termination. So let me write. The first termination we call that row independent termination. So let me write. Or we call that intrinsic termination. Okay, the second we call that row dependent termination. Now what's going on between these two termination? In row independent termination, as the name indicates, row is a kind of helicase like protein which acting as a helicase. So it is a kind of protein which is acting like a helicase, and due to this protein it can remove or it can drag out the RNA from between the two strands of the DNA. Okay, so whenever we need the row protein for the DNA for the termination of DNA transcription, then it is called row dependent termination. But when we don't need the row protein in termination of DNA transcription, then it is called row independent termination. Okay, now remember this one, row dependent termination, is very fast, or it can remove, or it can remove out, or it can release out or drag out the RNA strand between the two DNA strand very fastly as compared to the row independent termination. Okay, now what happened between these two? Okay, let's first of all explain the row independent termination. In this case, if you guys remember, we talked that, let's suppose this is the DNA strand. So all of you better know DNA strand are the double helix, but somewhere at that specific position, as you can see, the DNA are open. Okay, now let's suppose this blue region, as you can see, this is the promoter site, and this red one region is representing the +1 site where the RNA are synthesized, our RNA starting point. And let's suppose this is the RNA polymerase 2, which are synthesizing the RNA strand. So this will be the RNA strand which are synthesizing. Okay, RNA strand. Okay, now remember, when the RNA polymerase is moving in this direction, remember RNA polymerase is not always moving in this direction. RNA polymerase is also moving in this direction. Okay, they just need 3' end. So if we see the RNA strand, DNA strand, let's suppose this is the DNA strand. One strand have 5' to 3' end, while the other have 3' to 5' end. So we talked that RNA always use the lower strand, which is acting as a template, but the upper strand is coding strand. But remember, at 3' end, the RNA start their process. RNA polymerase start their process at the lower end, but at right side, from left to right. But at the upper strand, they can also start, but from the upper strand, they will again start the synthesis of the RNA. Now, the RNA polymerase from the upper strand will moving in this direction. Okay, and at the lower end, it will be moving in this direction, left to right, and from the upper strand, it will going from right to left. Okay, now as this is the RNA strand which is going in this direction, so definitely they will use the lower strand as a template. So this is the RNA strand which is synthesized, and these are the DNA strand which are present. Remember, at somewhere of the DNA strand, as you can see, somewhere of the DNA strand, there are a lot of addition of adenine. So means at that specific point. So let's a little bit enlarge that one area. Okay, so let's suppose this is the DNA strand. So at that specific position, as you can see, there will be a lot of adenine present. So we call that polyadenylation region. Okay, so this is called polyadenylation region. So from that position, let's suppose if the RNA polymerase reach from that position, this is let's suppose the RNA polymerase. Okay, now when the RNA polymerase reach at that specific point, then definitely they will add here uracil. So uracil will be added here. Okay, and between them a very weak bond will be formed, because all of you better know between the adenine and uracil there is a double hydrogen bond, which is actually weak hydrogen bond. Weak double hydrogen bond are formed between them. Okay, and somewhere of this RNA strand there is a GC rich region are present, means a lot of guanine and cytosine sequences are present in the RNA strand. Let's suppose at that specific position. So let me write GCCCC CGGGG. Okay, so these region are called as GC rich region. GC rich region. Now, as you can see, these are actually the palindromic sequences. Okay, so there may be a chances that these sequences can make a bond. These sequences are complementary to each other, so they will make a bond with each other. So remember, when we get like that structure, and when we get the palindromic sequences or at GC rich region, then it will make a bond with each other. So it will make a structure like that. Let's suppose this is the DNA strand, as you can see. Here is the adenine. And this is the uracil. So remember, at that specific position, a loop formation will be occur. So this is the formation of loop, and we call that hairpin loop structure. Okay, hairpin loop like structure. Hairpin, which the female usually use it for their hair, to tie their hair. Okay, means like that. So as you can see, this is the hairpin. This is the hairpin. So female usually use this hairpin for their to tie their hair. Okay, now this is the formation of the loop between of these two sequences. So these are the C, G, G, G and G, and these are the G, C, C, C and C. So that's why the loops are formed. So remember how the loops is created. Loops will be created when these two when the GC rich region are make a bond with each other. So how the bond will be formed? Because here these are the palindromic sequences. So when inside the RNA structure, inside the RNA strand, if the palindromic sequences are found, due to this way a loop will be formed, which we call it hairpin loop. And palindromic sequences in this case are the GC, which we called it in the RNA strand. These region are called a GC rich region. Why? Because here a lot of guanine and cytosine bond nucleotides are present here, due to which a hairpin loops are formed and by these palindromic sequences. So as you can see, hairpin loop structure are formed. How? Because they can come closer to each other. Remember, when they come closer to each other, these two region, when they come closer to each other, then what actually happen? There will be a force or a tension created on this one area. So this area is called UC. So this area is called as uracil and adenine. Uracil, polyuracil and adenine DNA RNA hybrid. RNA hybrid. So it means when at that specific position, means at polyuracil and adenine DNA RNA hybrid. So this is called polyuracil adenine DNA RNA hybrid. At that specific point, what actually happen when the loop is formed? So whenever the loop formation are happen at that specific point, then at that specific area the force will be created. So due to this way, it will be drag out. So whenever the loop is formed, so that area will be pulled. So when that area will be pulled, so this RNA strand will be drag out. So how? Because why the RNA strand will be drag out from that area? Because here a very weak bond are present, as you can see, double hydrogen. But at that specific point, we have the triple hydrogen bond are present, which is the strong bond. So remember, guanine and cytosine have a strong hydrogen bond. Due to this strong hydrogen bond, they will put a pressure and they will put a force on this area, due to which they will drag out the RNA strand. And once we get the RNA strand, so it will be look like in this form, a hairpin loop like structure. So this is called RNA. This is actually the RNA structure. So we will get RNA in this form, which is drag out. Okay, remember that area is also called, this one area is also called intrinsic signal. Intrinsic signal. Okay, so due to this intrinsic signal, when the intrinsic signal is given, then this one RNA strand will be drag out from between the two strand of the DNA. So remember, this is the two DNA strand. Okay, this is how the RNA strand are synthesized, and this is how the termination of DNA transcription without Row protein. So that's all about the Row independent termination of DNA transcription, or we also call it intrinsic termination. Now, why we call it intrinsic termination? Because these one areas, as you can see, which we call a GC rich region, it give intrinsic signal. It means that due to this way, the bond formation, triple bond formation, which is the strong formation, are happen, and it will drag out or it will pull this strand from between the two strand of the DNA, and finally we get the RNA strand. Now let's explain the row dependent termination. So let me a little bit erase it. So what happened in a row dependent termination? So again, in this case, we need a row protein. Row protein structure will be in this form. And they have two domains. One domain is called N, while the other domain is called C. This is row protein. This row protein usually need energy from ADP plus pi. So they give ATP, and this row protein use ATP to drive, to move. Okay, and it usually get the ATP energy from ADP plus pi. This is actually ADP. Okay, after that the row protein will bind and will cut the hydrogen bond between them. Now what happened in this case? Again, in this case we have the DNA strand. Let me draw. And that's why the DNA, remember, the back side of the DNA are become coiled. Okay, because that our replication bubble is now moving. Whenever the replication bubble is moving, then from the back side of then to the back side of this replication bubble, the DNA strand are again coiled. But during replication bubble, the DNA strand are uncoiled or unwind. Okay, now as the replication bubble are not replication bubble, transcriptional bubble, sorry. Now whenever the transcriptional bubble reach at that specific point, so then it will again open that one area of the DNA strand, as you can see, and while the remaining area are coiled again. So again, this is the RNA strand, this is the RNA polymerase 2, as you can see, having very large structure. Okay, and this is the RNA strand which are synthesized. So remember, sometime what actually happened in this case, sometime there is also, the intrinsic signal is always present at that specific point as well, but it doesn't mean that they will drag out by itself the RNA strand. Now sometimes they don't have the ability to drag out this RNA strand between the two DNA strand. Sometimes it is very difficult. For that purpose, the row protein is used. Okay, now sometime what happen, let's suppose these are the adenine, 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 as you can see. And here is the uracil, 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 uracil are present. So at that specific point we have. Now sometime what happen in this case? In this case, the RNA have a specific site which we called it a RUT site. Okay. So when the cytosine are present in a large number, in a huge amount, when the cytosine are present in the DNA strand, and when the guanine are present in a very low amount, let's suppose if we have guanine 3%, and if we have cytosine 70%, then this one area is called as RUT. So as you can see, that specific area is called as RUT. So we called this area RUT. RUT means row utilization termination site. Row utilization termination site. So it means that usually the row protein are bind at that specific point. Okay, so the row protein are bind here. So it contains 6 protein because it acting as a helicase. And all of you better know helicase can unwind the DNA strand, and they can also break the hydrogen bond between the two strands. Okay, so this is acting as a helicase, and this is the Row protein, as you can see. Now this is the C site. So the Row protein are loaded here. When the Row protein are loaded here, then it is moving around this strand. So it is moving in the strand, as you can see, and reach at that specific point. So let's suppose this is the Row protein, as you can see. So whenever the Row protein reach at that specific site, then what actually the Row protein doing? It will break the hydrogen bond between the adenine residues and uracil residues. Okay, so it will break the hydrogen bond between them. As hydrogen bond between them is already weak, so due to this way, it will easily break the hydrogen bond fastly. Okay, so when the hydrogen bond, so remember, when the hydrogen bond are break down between them, so again the strand will be released out and we will get the strand in this way. Again we will get the strand like this structure. So in this case we need this Row protein. Okay, so let's suppose this is the 2 strand of the DNA and this is the RNA strand which are synthesized. So here, as you can see, between them there will be the hydrogen bond. So this is, let's suppose, the Row protein. Row protein will reach here and they will, break the hydrogen bond between them, and then they will remove the RNA strand between these 2 strands. So when the RNA strand are released out, so definitely the RNA strand will be modified just like that, and we will get again a hairpin loop like structure, because again we better know that there is the guanine and cytosine rich region are present. That's why it make a hairpin loop like structure. This is all about the termination of DNA transcription in prokaryotes. I hope you guys get the idea about the termination of DNA transcription in prokaryotes. But if still you have any kind of question in your mind, you can write it into a comment. So if you like these videos, make sure to hit the like button, share these videos, and also subscribe the channel to get more interesting videos like that. Thank you so much.